My co-hosts today are Felix and Giovanna. They are my co-developers for the package we're going to be using, Spectre. And we're going to go through a bit of an introduction to using R for computational analysis, which is kind of the, I guess, the foundation of, what we do for a lot of our work, using computational methods to help us understand things about the immune system. So I'll hand over to Felix/Giovanna, and we'll get started. Who's starting?
Giovanna.
Cool. Do you want to use? You can use your. Yeah, it's fine either way.
Right, can you hear me? Can you hear me? All good?
Yeah, that's good.
Okay, so now I'm just going to share my screen. Oh, hi everyone. Hopefully you can see the slides okay. So today I'm just going to give a little bit of a presentation first, sort of introducing everyone to R in RStudio, just briefly how to install them, what packages are, what are functions, which is essentially hosted within a package. And then after that, I will go through a quick demonstration on doing some basic data manipulation using R and Spectre. And then after that, we'll give about an hour or so to everyone to play around with the some codes, basically the session 1 script that Tom have uploaded, and yeah, and ask questions, of course. So let's start with that. So traditionally, as everyone probably more familiar with analyzing data set, is normally using Flojo, which is just software. The alternative to that is basically using programming or code. So there are so many different languages in the world for programming. There's Python, there's R, there's C++, so on and so forth. You can treat each of these programming languages as just like a normal human language. So maybe R is in English, Python is German, something like that. And in this session and the subsequent session over the coming 2 days, we're just going to be using the R programming language. And often when you start analyzing data using code or programming, if you talk to other people, they'll be asking, oh, why don't you use Python, or why don't you use XXX programming language? At the end of the day, my take on this is that, look, all these are just simply tools to analyze your data set. There's no right or wrong answers as to which language to use and what not. Use what you think is the most comfortable for you. And of course, pick a language whereby the packages and all the libraries, which I'll go to later, are more available for your purposes. So what is R? So I've explained what R is, but what is RStudio? I think you probably have heard of the software called RStudio many times. It's simply just a software for you to write a code and to run R code. So you don't have to use RStudio to write an R code or to run R code. You can simply just use a normal text editor, like Sublime Text or the normal text edit on a Mac, and then run the R code using a terminal or command line. But it's very complicated to do that. I don't highly recommend it, especially to people who are not familiar with programming and stuff. And that's why a lot of our tutorials for today and subsequent days will be using RStudio. They're freely available on both Mac and Windows. And within RStudio, you have the text editor and a few functionalities to run the output and so on and so forth. Okay, so the next week will be how do you install R and RStudio on your computer? Yes, we provided some virtual machine for these particular sessions, but I think it would be good for you to go home to be able to install them yourself on your personal computer, just in case you want to use it. Oh, I hope you do at the end of our sessions. So the R can be downloaded from this website, crancsi.au, and the RStudio can be downloaded from this relevant website. There'll be an option for Mac and Windows. You just have to pick which one is relevant for your computer and then just download it from there. I can remove this. Sorry, I'm just removing my mouse so it's a bit easier. So this is what RStudio look like. You can have a different color scheme. This is a dark color scheme, and you can also have like a white color scheme. There are a few different panels. The first one, the top left, is called the text editor panel. So this is where you type only your code. And then you also have the bottom one in here, which is a console panel, I call it. So when you run a code, so with RStudio, you don't have to just run one script or one file. You can just run each code line by line, as you'll find out later on. And as you're running each line of code, you can the console panel will probably tell you whether everything is well. And if it does, it just show you the chunk of code that you just run. And if it's not, it will just simply tell you there's something wrong with it and what is the reason behind it. The panel on the right-hand side, top right, basically have a few different tabs. The first one is called Environment tab, which basically tells you all the variables that you have currently stored within RStudio. So I'm going to go through a little bit what variable is. So you can think of variable as a container to store your data. I always use analogy of a dish. So you can think of your data as a food. The plate is basically the variable that hold the food. And as you type your code and loading in data and all the stuff, you find yourself growing more and more variables. You can have more and more variables over time. And it's also worth noting that variables can be of various types. So for instance, you can have character, which is basically just letters, words, sentences, or it can be a numeric, which is numbers, or it could be logical, a type which can only either be true or false, or it could be more complicated type, which is say a vector, which is basically a collection of any of the above, like character, numeric, or logical, or a list, or a data frame or data table. Later in the demonstration, we'll go through each of these in greater detail, and you'll probably get a better understanding and appreciation for them. It doesn't work. Oh yeah, here you go. All right, so that's the top right. As you can see, there are also other other tabs which are less relevant if you're just starting. And I personally don't use it myself too much, honestly. The bottom right is basically a few tabs as well. The first one is just simply a file manager. It looks just like file manager basically. And the second tab is basically what I call a plot viewer tab. So for instance, if you draw some kind of a diagram or plots, you can actually view it in this screen, in this panel, sorry. And the next one will be like packages and help, which I'll go through next. So first of all, what are packages? You can think of packages as a bunch of reusable R codes. And within packages, there are basically functions, which basically are the reusable R code. Each function is responsible for doing something, generally doing only one thing. You can think of a package as a recipe book and a function being a recipe inside the recipe book. And Spectre is basically an R package that we develop. So back to the RStudio layout. So the packages tab basically shows you all the packages that you have installed in your system. You can click on any of these to see what each of the packages are. So for instance, one of these will be, say, a base, which is an R base package that comes with your R installation. And here comes the trickiest bit of all, in my opinion, when you want to use R to analyze your data, is that how do you install these packages? And unfortunately for R, we have 3 different channels of distributing packages. First one is from a source called CRAN, which is what most packages are hosted on. Second one is called Bioconductor, which is more relevant for a lot of tools, computational tools that are generally used for biological data set. And also from Github, which if people who are more familiar with programming, you know that Github is where people store code and share code for particular packages and so on and so forth. And say if you're given a particular package, how do you know which source you need to install the package from? The first thing to do is to just simply Google the package name, it should tell you. Or alternatively, the manuscript that describes the package should actually tell you where to download it from. And of course, different packages require a different way of installing. And we will basically go through that later during the practical session. And the next one, I'm just going to give a brief introduction to what Spectre is. Spectre that we developed basically contain various computational tools that is designed for exploring and analyzing single cell data. There's a DOI here that is a link to our publication that you feel free to download and have a read if you like. What Spectre has is basically a bunch of functions that allows you to do data pre-processing, such as the arc-sign transformation. And it also have the ability to do batch alignment using a algorithm called Cytometry. And it also provides the facility to do clustering and dimensionality reduction. So using flowsum, phenograph, using Tisney and UMAP. And it also provides some functionality to actually do some quantitative and statistical analysis. And all of these will be clearer to you in tomorrow's session, which will go through some clustering, dimension reduction, and some quantitative analysis for some data set. Next one, I'll be going through some basic data manipulations in R Spectre. So this will be predominantly some demonstration that I'll perform later. But before I get to that, I'll probably briefly explain an anatomy of a function. So in the previous slides, you probably heard me talking a little bit about, functions and so on and so forth. Function being the tools that are within a package. So each package contains various functions, and each function has a name and sometimes a parameter. And this parameter basically governs how the function behaves. So for instance, if you run a clustering function, it may have a parameter that allows you to specify how many clusters you want to produce. And if you are familiar with Tisney, you often hear the term perplexity and so on and so forth. And those are basically parameters. And a function may return a data, which you can later on store in a variable, or it may not return anything at all. So this is what a function would look like. So this is the name of the function, and these are basically the parameters. And next to each of the parameter name, there's basically like an equal sign that is like an assignment sign, and you can assign each parameter with a value. So for instance, in this function, which is built to Spectre called read files, what we're telling it is to basically read all the files in a particular directory, but only just read the FCS file. And we'll store that in a variable called DAT. This is something that is worth noting. You can't expect individual packages to not share the same function name. So for instance, we have a function called read file in Spectre. Maybe in another function, there is also another one called read files. And so when you want to tell us basically which packages to get this function from, you can prepend the actual package name beforehand. And so, so this is what I'm going to do next. So a little bit of a demonstration on how to create and save, write R script, run some code, setting working directory, loading packages and installing packages, and then reading some FCS, or not the CSV files, investigates the data, and then finally export the data back out as FCS and CSV. So before I move on, is there any question that you like us to answer before I move on to the demonstration? You can either unmute yourself or just throw it in the chat. One or the other will do. Cool. If not, then I'm just going to move on with a quick demonstration. So I'm not going to use the virtual machine because I'm more familiar with the my own Macintosh. So this should be the session one script that you can download from the link that Thomas provided in the website, in the email. So first of all, the very first one would be, how do you install a package? So there are a few ways of doing it, as I mentioned before. Before I move on to that, maybe it's a good idea for me to explain what is an R script. How do you create one and to save one? So if you want to create a new R script, you can go to File and then say New File, say R script, like that, and then you can type whatever code that is that you want to type within your script, and then you can do a save, give it a name, and whichever directory you want to save it for, and then just click save, basically. So that is that. So if you want to run an R code, one way of doing this is like for instance, I'm going to create a just a variable for sample variable called say So if I've typed some R code and then I want to run it. So what this one I've just typed is basically I want to create a new variable, call it example variable, and store number 1 into it. What you can do is basically either highlight all of these, and if you're on the Mac, you can place a command enter that will basically run the code. And as you can see, after I run it, you get a new variable inside the environment tab here. Or you can either say code and, and then you say run the selected line. And I think if you're on Windows, you can press Control Enter to run the code. So this is how you run a code line by line. And if you want to run the entire R script, you can either select everything and then say run selected line. Or alternatively, you can just simply go back to code and then say source. What it will do is that it will just simply run everything inside the script. So that is that. Now, I might quickly just demo what is inside a packages tab in here. So for instance, I have a lot of packages installed, and I'm just going to show what a help file for a particular package look like. So for instance, this is the Spectre package that we developed. If you click on it, it will show you something like this. I hope it's quite clear. I'm just going to drag it up so it's a bit bigger. So this is what we often call like a what we call it, reference file kind of thing. It's like to help pages to basically describe what are each of the functions that are available in Spectre. So for instance, we have a create detail. We have the one that I put on the presentation before called read files. And if you click one of these, it's a function. It will tell you what is this function is supposed to do. And it will also tell you how to use it. What are each of the parameters or arguments is a synonym for parameters that it has, then what sort of value that you can give it and what does each parameter mean. And then it will tell you whether it returns something or not. In this case, we just return a data table. And sometimes at the bottom, you see an examples, which will tell you how to use this particular function. So that is that. Sorry, I'm just trying to minimize this so that I can go back to the text editor. Yay, here we go. Right, so that is just a quick layout of the RStudio. It will look similar to the virtual machine or on any Windows machine or Mac machine, to be honest. So the next one I'm going to go through is installing packages. So as I said before, there are 3 different ways of installing packages, either from CRAN, from Bioconductor, or from GitHub. So this is the I would call it the syntax. How to install packages from CRAN is basically using a command called install.packages, which should come with your base R installation. And within it, you have to pass it the package name as a parameter. And then sometimes, funnily enough, you need to tell it what repository that you want the install function, the install packages function to look at. To find this particular data table package. Sometimes you need it, sometimes you don't. If you need it, it will tell you when you run the install packages that, hey, you need to specify a repo. If you don't need it, then it will just simply install it for you. You can install it like this using code, or alternatively, if the package is available from CRAN, you can just go to packages here, the package tab, and this install button. And then you can search for the packages. Say, for instance, I want data table. And as you can see, as I'm typing it, try to sort of tell you what are the packages from CRAN. And then you can install it. You can probably get this to look at the Bioconductor repository, but I haven't tried it myself. So I wouldn't, Anyway, so if you want to install it, then just click the name of the package and then just click install. I already have data table, so oops. Don't worry, it will happen a lot. So for instance, if you try to install a package that is you already loaded, and that is the case in my screen before, what will happen is that it will tell you that you need to restart the R session for R to basically reinstall it for you. It's no dramas. The key thing about all of this is that if you find an error, don't panic and just simply look into what has happened, and yeah. So anyway, after you've installed it, it will tell you whether it's successful. In this case, it is, and there's no obvious error message. And you can actually double check whether it's been installed by just going to the packages here and look for data table. And if you find it, boom. Sometimes after you've installed packages, you probably have to restart the RStudio or the R session, and you can do that by simply go to session and say restart up. But just bear in mind, when you do this, everything that you have stored before in the environment will be gone. So just bear in mind. And next one will be installing from Bioconductor. So Bioconductor, for installing Bioconductor packages, you need to install this package first called Bioc Manager from CRAN, and it's the same syntax as the one before, except package name for Bioc Manager. After you do that, then you can use that Bioc Manager package and run the install function, and then you supply it with the package name. So for instance, in this one, I'm trying to install Flowsum, which I should already have. It'll take some time. Yeah, sometimes something like this will occur, like it will try to install any other Bioconductor packages. They install a newer version of the Bioconductor packages that you already have. And you can either update all of them, some of them or none. I generally choose none just because sometimes updating packages can result in breakages in some of them, unfortunately. So I tend to play safe. Unless I really need it, I won't update it personally. This is stuck. Whoops. I'm gonna try again. It's stuck somewhere. Say none. And that's it. Generally, it just if it tells you a warning message, you just read it, and if you think, oh, it's all right, then just leave it. Don't need to worry too much about it. In this case, what the warning message basically said, look, you've already got a Flowsum installed, and I'm just not going to install it again. The next one is from GitHub. So GitHub, to install any packages from GitHub, you need to first install this package called remotes from CRAN. And then use the install GitHub function that is built into remotes package to install the GitHub. So this is not as straightforward as just putting a package name, because the thing with GitHub is that you can probably have a similar or the same package name, but it's just hosted by different people. So in this case, you need to put in as well which people own the repository for this package, and then followed by the package name. So for us, it will be Immune Dynamics team own the Spectre package in GitHub, and then the package name is obviously called Spectre. So yeah, if you run this, it will download the Spectre package and install it. I'm not going to run it because it will take some time. So I'm just going to move on. So that is installing packages. The next bit I'm going to go through is basically setting a working directory. Working directory is basically just a default location within your computer that your current R script will be running from. And when I say running from, will be like, e.g., when it's trying to load some data, it will first look into this particular working directory to see if data set is there, or exporting that. It's like a default directory to do anything, basically. And you can set a working directory using the command called setwd, and then you just simply supply whatever the path to the directory is. In this case, this directory is basically stored within my computer under the documents folder. And if I do that, and if I do this function called list file, this is another trick that I didn't tell. So if you want to write a R function or particular code, but you don't want to include it in this script. So sometimes you just want to try things out. You can just simply type it directly into the console like this. So in this case, I want to list all the files that are within my working directory. And as you can see, there's quite a few in here that will correspond into whatever is within my documents directory. And one way to find out if you have done a lot of manipulation, you don't know what your working directory is, you can run this getwd function, which will basically tells you what is your current working directory. Hope it's simple enough. And the next one is called dir.create, which basically will just create a directory within your working directory. So for instance, if I run this, it shall create a new folder called the ACS workshop, which was not there before. And of course, you can set your working directory to this newly created directory. And when I run get WD again, it should tell you that I've changed the working directory to the new one that I've just created. I hope that's simple enough for everyone. Don't worry about all of this. Just pay attention, and then we'll give you some time to try all this code yourself later on. So the next one is, say, for instance, you have installed the packages, right? How do you load them so that you can start using the function? So all of these you can run using, you can do using the command called library. So in this case, I am loading the Spectre packages like that. And you notice that if the package loads successfully inside this tab, packages tab, if you type Spectre like that, oh, that checkbook's missing. It should be tick, basically, to sort of indicate that you've loaded it. Let's maybe try to load something else like data table. Like that. Don't worry about that. And if you scroll down to, or just do search data table, all right, it's normally tick, basically. I don't know why the checkbox was missing on my computer. Sorry about that. But generally speaking, all this checkbox will be ticked after you loaded the package. This is so strange, but you know, this thing happens. Anyway, don't worry about that. So that's how you load a library. It still works, don't worry about that. And the next one is how do you read, say, a FCS file or CSV file? So for this, I'm just going to use Spectre. So looking into my directory, this is empty. I'm supposed to fill it in with the data set, the demo data set. Whoops, just give me a second. It's in a separate directory. Pardon me. It will take a minute. There you go. So let's just assume that the my working directory already have all the data set that I need to load in. So if you go list files, you should be able to see all these CSV files, which is these are basically just the demo data set that we have made available on Spectre. You should have all these files in the directory that Tom has shared in the email, so you can download them and save it into your working directory. And you can use this read files function from Spectre, which I've explained before, to read in all the CSV file into a variable. So let's just run that. And when it finishes, it should actually store a DAT variable in there. Let's have a look what the DAT is like. And you can do that by either clicking on this magnifying glass, and it should open a new tab and tells you what is inside this variable. Alternatively, you can also just put the variable name in here and say DAT, and it shall give you some sort of a preview as to what is within there. This is what we call a list data structure. So you can think of it as a bit like a what I call it, an array. So it's like a collection of, in this case, will be a data table, which is just collections of tables. And in this case, each element in the collection is basically one CSV file. And sometimes if you want to check what type of, what is the type of the variable, you can just use the function called class, and it should tell you what it is. And if it is like the previous one that we have the example variable, like this one here, if I type class example variable, whoops, she'll tell you it's a numeric because it's a number. And if I say, so this is not a trick. If you're on the console, you press up key, the arrow up on the keyboard, it will show you all the code that you have run before. So if I change this to say example variable to A, and if I run class again, it shall tell you it's the type character. Simple enough. This function called names will basically just tell you what are the name of each of the element within the list. It's actually quite handy, but depending on what is the type of this variable, it will tell you different things. So for instance, if it is a data table, it will probably tell you what are each of the column headers in the data, in the table, basically. It'll be more obvious later as I progress along. So each of these, this DAT that we just read in is basically a list. Now, I don't want a list. Generally speaking, it's a bit easier if I sort of process and manipulate the data if it is just one giant table containing everything. And that's why in Spectre, we have this thing called do merge file, which is basically collapse the element of all the lists into one giant table. You see what I mean when I run this. So if I open that again, so instead of a list like this, you get a one giant table here that everyone is probably more familiar with. You can see there's a NK1.1, that is CD3, CD45 marker, and so on and so forth. Each of these columns will correspond to one column within that CSV file, basically. And what we've done is basically just combine them all together into one giant one. So there's that. And if you check the class, it shall tell you it's a data table. Or in this case, it's data frame. So a bit of history about this is a data table is like an enhanced version of data frame, put it that way. It's easy to use. So that is loading in data. So the next one will be just investigating the data, sort of looking into what is within there and so on and so forth. I mean, you can actually just, open this and it show you the table, but if you have a lot of data in there, a lot of cells, you don't want to be sort of keep on scrolling to find something. It's not what you should be doing. So what you can do is that I'll show you a few functions to sort of start investigating, manipulating some of this, just a very simple one. So the first one is called a head function, which will tell you the first top X number of lines in your data set. So the X is basically given by this parameter called N. Sorry, I should have used N. So if I change this N from 10 to say 20, what it will tell you is what is the first 20 row inside the data set, inside your data table. And as I mentioned before, if you use this function names, it shall tell you, in the case of data table, all the columns that are within your data set. And say you want to save some of this, say you don't want to keep on running this, you want to save the list of markers or the columns into a particular variable, you can just simply do this. So basically call names that function, and then I just want to keep element number 1 until element number 9 into a variable called marker calls. So let's see what happens when I run that. You see that instead of saving every single column name into a variable, it will save just the first 9 columns, which is all the way from NK1.1 to CD4. So we exclude these 2. And if I change that to say 8, you see that CD4 will be missing from marker calls. There you go. Simple enough, I guess. So the next one that I thought might be useful would be like, say we want to subset the data. Say so our original data have like this file name and file number column. Say we don't want them. Say we just want the markers. So what do we do then? So we grab all the columns that contains the markers. So we rerun this again. Just check that it contains all the markers that we want. And you just run this particular sort of function from data table, which basically telling it, I just want to keep all the columns that are stored within the marker calls a variable into a new variable called that marker only. You see what I mean when I run it? And if I open that, you find that we no longer have the file name and file number like the one we have in the original DAT variable. We only have all the markers. And say, let's pretend we have forgotten how many samples we have or how many files we have. And like, put it this way, say you want to put this file name column back in. Say you have forgotten to put it back, you meant to put it there anyway. And what you can do is that you can use this dollar symbol, which basically tells data table to just isolate the file name column, and we'll store it into the samples variable like that. And let's see, okay, it contains all the file names. And what you can do is just call DAT, we'll give it, I don't know, file name. We'll append it to the dot marker only. Let's just call it new file name, something like that. And take it from the samples variable. You'll see that when we look into the variable that we created before, you find a new column called new file name that is basically taken from the file name column in the original data set that we loaded. Another one that I thought would be wait a minute. Something.
So,
Oh, good. Yeah. Okay. The other thing that I thought was, sorry about that, interruptions. So the other function that I thought might be useful is this function called unique. And as the function name already sort of allude to, it will basically try to find a distinct isolate just a distinct element within, say, a collection of elements. So if you imagine that collection, or should I call vector, that's probably more appropriate, is called samples that we have isolated before from the original data set. If you run the unique function on those, it will basically just tell you all the unique samples and all the unique file names that are within whatever that vector is. You can use it on any columns of your data set. So for instance, if I just run the unique, I just want a unique file number of my data set, it will just simply, you can just run unique and give it the column, the content column of your data set. And then the other one I thought might be interesting would be, say for instance, you want to run a commonly used stats like mean, median, and so on of any particular column in your data set. In this example, say we want a mean of a CD3, you can just run the mean function that is built into the base R package and supply it with the CD3 column of your data set. So if I run that and have to look at the CD3 variable, it should tell you the mean value of the CD3 column inside your data set. And this one, you can change it to median and it shall give you the median instead. Like that. As you notice that as I'm typing things like, say I want to see standard deviation, SD, I think, like that. Sometimes this kind of pop-up will show up and it will basically try to help you understanding what are the parameters that is inside there, what you need to supply. I found it quite handy. And also sometimes as you're typing a function name, it shall give you, it should try to auto-complete it for you, give you some sort of suggestion that I thought might be quite useful as well. So yeah, that is that one. And say for instance, hypothetically, you want to have a new column that is basically a summation of CD3 and NK11. You can just simply do, not this one, you can just simply do something like CD3 plus NK11 be equals to whatever that CD3 column, so DAT CD3 plus DAT NK11, like that. So what it will tell you is that it will sum up the CD3 and the NK1.1 expression of individual cell, and it will store it inside a vector, something like this. You can see there's a lot of it, and you can just assign that into your into the original data set using something like DAT and give it a column name, say CD3 plus NK11 equals to whatever we just worked out before. And if you look at the DAT, the original data set, now you'll find a new column, which is basically the sum of CD3 and NK1.1. That's easy enough. Alternatively, if you can do it that way after you storing it the variable and reassigning it, or you can just simply do what I call appending it as a new column using a data table notation. So something like this. Say I want to add Ly6G and CD11B. So I'll call the new column Ly6G plus CD11B equals to, sorry, I forgot the comma here, Ly6G plus CD11B, something like this. And if you go back to DAT, you find a new column, which is basically a sum of Ly6G and CD11B. So that's just a simple manipulation. say, for instance, you want to inspect just the isolate. This is quite common, I found. Cells that have a positive expression of Ly6G and positive 11 of B220. You can run something like this. So what this is telling data table is basically look into the DAT variable and only isolate cells with Ly6G greater than 0 and B220 greater than 0. So if I run that and look into the content of DAT subset, it will basically only show you cells with positive Ly6G and B220 expression. And these are just some stats you can run. So for example, on a minimal, the cells with the lowest expression of Ly6G, you can just run that. It should tell you. It should be different from the original data set that you loaded in. So if I find the minimum expression of Ly6G on the original data set, you'll find that there's one that has a negative expression, which is no longer included. It will be the same for B220. There. So then the next one, the last one actually I want to go through is, say you have done some manipulation, you want to save it out as a CSV file. How do you do that? Spectre provided a function called the write files, which will basically take a data table, any data table, and write it out as a CSV file. Say, for instance, you've forgotten what a write file is, or how does it look like. You can look into the function in here in the Spectre package, so you can scroll down and then find write files. Or alternatively, the shortcut will be like, if you remember what the function name is, you can type a question mark followed by the function name like this. And what it will show you is that it will basically pop up in here the guide for the write files function, or whatever function it is that you want to query. And so let's just try to write that that subset data into a CSV and an FCS file. So with Spectre's write FCS function, you need to install the Flowcore package from Bioconductor. So let's just install them, and you got to load them obviously. And then you can run the write files function like this. File prefix is basically telling the function what is the file name, and write CSV or write FCS basically true or false, whether you want to write CSV or you want to write FCS file or both. Like that. That didn't work. Let's load Flowcore again. Normally when that happens, it's because I didn't load Flowcore well. And that should end up with a CSV file like this and an FCS file. All right, that's going to take forever. I'm not going to load it. Yeah. So that's the end of the demo. Is there any questions? Because we just simply from here, feel free to sort of play around on it with your computer or on the virtual machine and just unmute yourself to ask questions.
For anyone who's still keen to use a virtual computer, I'm just going to drop those links back in the chat if you still want to hop on. Otherwise, feel free to tackle it on your own computer. Please feel free to give it a shot and ask us questions as you're going. If you want us to go through something specifically or kind of, I suppose, demonstrate some of the aspects again, we're happy to do that. But this is a good time where you can tackle it, give this kind of stuff a shot, and then we're here to help if there are issues or questions. Oops, it doesn't actually put the URL on there. It's not helpful at all. think that tick box thing, I think it was like, it's actually ticked, but it's not rendering the correct graphic, so it's just disappearing. Yeah. It's really like, it's really weird.
Yeah, it's like normally it just.
Yeah. That's usually the solution. Just going to try and see if we can get the speakers working again. So brace yourselves. Sounds okay. I don't think it accepts microphone input from the speakers. I think it just does the microphone from the laptop.
My checkbox is full.
It's very strange. Okay. Is anyone running the code successfully and getting this to look good on their system? We'd love to see how things are going. I'm also aware a couple of these accounts are like multiple people in a room. We'd love to hear how you're going and how the conference has been so far as well. Excellent. The other thing to mention is if anyone is at the stage where I don't know, they've kind of done this and they're like, all right, I've got a good feeling for running the R code. The link that I dropped in the chat for that getting started covers all the basic stuff. The sessions in the subsequent workshops are both online as protocols that we've got on the Spectre website. So if you're keen, you can go and dive into any of those as well. They have like data sets that come with the scripts and full worked examples, and you can kind of move through that as well in your own time. So you can access those at any point. It's hard to tell whether it comes through there actually. No. We thought we'd also mention, if you're interested in installing these things on your own laptop and haven't done it before, this is a good chance to give it a shot. For some reason, installing packages is just the worst part of this whole process, and there's always little problems with it. They're not insurmountable, but they can be challenging. So this is a good chance to sort of tackle those. And if you give it a shot and something goes wrong, let us know. We can help you walk through it. If anyone is working on, well, I'm sure everyone's working on something cool, but we'd also love to hear from you about stuff that you're working on as well. If you feel like sharing, don't feel any pressure as well. But we love hearing what the attendees of these workshops, are doing with their kind of analysis work. So please do feel free to share with us if you feel inclined. So I hope everyone's being able to get some progress with their R scripts. Again, this is something you don't have to necessarily do right at the moment, but this is a good chance to kind of run through some of the script that we provided in that link for today's data set, get some interaction with installing the packages, interacting with the R code. And that's, I guess, the most important thing. Once the interaction becomes more familiar, everything else becomes pretty manageable. What I thought we might do, just to kind of, I suppose, round out a couple of these aspects, is look at what an actual experiment looks like when we're doing it kind of in real time. So this is good. So this is what we would do if we were going to say run an actual experiment, and this is actually going to be what we're going to tackle in the next workshop for those of you who are registered for that. But this material is also online on our Spectre website. In just in case anyone's not sure how to get there, Max, Spectre, and I'll put this actual URL in the chat so you can find it. So it'll be this page. Just drop that in there now so I don't forget to do it later. The place where we usually start is this one, the simple discovery workflow. Essentially, what we try to do with this approach is basically take in a bunch of data, do some preparation to it, perform some clustering, perform some dimensionality reduction, and then sort of do some basic statistics on the output of that data. The idea is that it's all in a single script in R, and so you can kind of do it all in one big hit. We've also got a version of this protocol where you can replicate these steps in Flojo. This type of approach doesn't have to be done in R. You can replicate this in other programs. The reason we like doing it in R is because it gives us a couple of things. One is kind of, it gives us a different way of scaling up our analysis, but also automating a bunch of it. What we find by using R is we can sort of manipulate the data or reconstruct the data, do different things to it in a way which is fairly seamless and very reproducible. If we wanted to, for instance, run clustering steps or dimensionality or steps in Flojo, there's a whole bunch of kind of downsampling, merging files, exporting them, re-importing them, things like this. These are all kind of manual labor steps. There's sort of lots of pointing and clicking, lots of processing, but it's kind of taking up your time as opposed to just giving the computer sort of the instructions for what you want it to do and letting it get on with the job. So if we click this link here, this is basically a big page which kind of just walks through what our simple discovery structure is like. It comes with a demo data set, which is some brain data, which if anyone caught Alana's talk earlier today, it's actually, it's not the same data, but it's very similar data. So cellular infiltration into mouse brains in a viral encephalitis model. And then you can basically just kind of follow the instructions as it walks through the whole process. I just wanted to give a little bit of, just a bit of an introduction to how this works in the real world. We end up with a script. So in this case, the way we structure our simple discovery workflow stuff is we have a folder and we put in there our data. Now, something that we didn't cover in kind of specific detail is how you actually get these CSV files. On the website, on that page we were at just before, we have a whole protocol here which talks about like initial cleanup steps and how you might export the data from a program like Flojo, but it could also be from Diva or from any other program, essentially. This allows us to kind of isolate just the cells that we want to analyze. There are, of course, a number of ways you can do this in R as well. By preference, we've organized it this way. I guess what it reflects really is a personal preference. I certainly like taking things into something like Flojo first, where I'm kind of messing with it manually before I then take it into a more kind of automated process in R. That's the way I guess I like to do things, and so that's kind of a reflection there. It's absolutely not the only way to do that. But to kind of get started, we have some prep instructions there. And then when you export the CSV files, you get something that looks a little bit like this. So we get a folder for data and a folder for metadata. There are some instructions on that page to talk about it, but this is essentially just so that we can have information that relates to each sample in a kind of a tidy format so we can import this easily. This is going to be important when we come to do things like our statistical analysis. We kind of want to know what file, what sample belongs to each group. We can also include all kinds of other things, batches, cell counts, whatever we want, whatever relates to each sample, we can add in here. If this was clinical data, this might include age, sex, it could include all kinds of different things and other clinical measurements. And then we have a folder here with our script. And when we open that script, it will look a little bit like this. So much the same as how Giovanna was explaining, we can kind of take this one step at a time. We run the little bits of code bit by bit, and that kind of helps us build up our analysis and kind of get to a point where we're running things at speed. So we're not going to go through the whole thing today, but just give you an idea of how this gets going. You can run all of this in your own time if you're interested. All of this is online. But much as Giovanna said, what we start with is, the way we interact is we can highlight either a section of code if we want to run multiple lines, or we can just click on a single line and then click run. These first steps, typically of all our workflows, they start with loading the libraries that we're going to need. So these are the actual packages that we're going to rely on for the analysis, which is very important. Once we've loaded the libraries, then we typically will move on to pulling the data in so that we're able to kind of get on with our analysis, so to speak. So this is something which we didn't, I don't think that script had the auto directory finding thing.
Auto directory.
Yeah. So this is something which I do like to bring up when introducing people to this. This is a really nifty little function here where the location of your script, as long as you put it in somewhere meaningful, the RStudio will actually figure out where the script is saved and save that as your working directory, which is really cool. It's just a nifty little trick, and if you're not familiar with setting directories manually, this is just really handy. So as long as the script is saved, in the correct location, so you've got a folder which contains data, metadata. This can be called anything, but we just call it Spectre Simple Discovery, and then the script there. As long as you're using that structure, all of this will work. Basically, this is just kind of follow the process. We kind of work through the bits, one step at a time. We set our directories, and then we will pull our data in at this point. Just to give you a brief overview, the steps are in most cases going to be very, very similar, regardless of what's actually being done, because the broad steps are always going to be, in common with each other. So, for instance, the very first thing we have to do is pull our data in, because obviously by definition we can't do any work without the data. So that will happen first, and then we're going to do some manipulation. Manipulation is not the right word. We're going to do some preparation of the data. For reasons which we haven't covered in detail today, but this is essentially to do with scaling, so that way the computer can see the data in a way which is meaningful, rather than seeing kind of the inherent noise that we get with floating cytometry data. So we do some preparation, and then down here we add the metadata and we set some preferences. I call it preferences here just because it kind of feels like a familiar term, but essentially we can tell the algorithm to do some things downstream. What that means is once we've got all these kind of preferences set, when we get to this stage, we're actually running the clustering and the dimensionality reduction. This is kind of where all the the cool fancy plots come from. These steps can take quite a bit of time to run. And so the idea with having this sort of preparation up front is, all the preparation steps probably only take a couple of minutes really to run through and to sort of make some decisions and run the code. Running the clustering may take some time depending on the data set. And so it could be 10 minutes or it could be a couple of hours, depending on exactly what's happening. And so at this stage, you can kind of run it, let it run, go away, go to lunch, run it overnight if it's a large step. After clustering, we almost always annotate. So we kind of get the data in, make some preparations for it, do the clustering, and then we want to label the cells that have been clustered so that we understand a little bit about what's happening to them. Having a group of cells called cluster 1 is helpful, but it would be arguably more helpful if we could say, okay, well, those are our CD4 T cells or our CD8 T cells. It gives it some kind of meaningful, interpretable answer. Once we've got the annotations, then all that's really left to do is to summarize the data. Essentially, this is quantifying various aspects of the data and then doing some statistical analysis on it. In this case, we do all kinds of metrics and we just calculate them simultaneously. So things like the proportion of cell types that we find in each sample, the actual cell counts that are present in each sample, how many cells are expressing a certain level of, one of a number of markers, all of these things that we would normally get in Flojo in, say, the table editor. But rather than having to sort of drag and drop various things out of a gating tree into the table editor, it will just calculate it automatically using this sum table function. Doing that means that we basically get a table which, instead of having cells as our rows and then the kind of the different markers as the columns, each row in this case will be a sample and each column will be something that we've measured. Number of T cells, frequency of neutrophils expressing a certain marker, etc. Once we've got that, it's reasonably trivial to just go ahead and make some graphs with some statistical analyses. So all of this you can find, as I mentioned, in this link. And you can see all the different types of plots and graphs that we create in this process. For any of you who are coming to the introductory session, introductory analysis session tomorrow, we're going to go through this kind of in detail, step by step, and actually run through that analysis. But just to give you an idea of kind of what we end up with in the clustering steps, we end up with essentially two things. This plot is a dimensionality reduction plot, and this is simply a technique that allows us to plot cells on 2 dimensions in a way that represents higher dimensional relationships. So in this case, if you'd imagine that each dot here represents a cell, and the 2-dimensional spatial arrangement roughly correlates to how related the cells are. Now, there's an important point to make here, which is one that we always need to bring up, is that these are not absolute. Like these distances are not absolute. Whenever we do dimensionality reduction, we have to make a compromise in terms of how we can get this information on 2D, right? And so typically the easiest way to think about this is that cells that are grouped together in, let's say, an island, we would consider that they're similar to each other. But the fact that the distance from cluster 8 to cluster 1 is kind of a long way, that doesn't actually mean that they're all that different. It's really only the local relationships. The fact that all these sort of cluster 1 cells are near each other, that's meaningful. The fact that all these cluster 8 cells are near each other, that's meaningful. The fact that these two have a lot of distance between them is not meaningful. That overall arrangement is a compromise for how we can get this data into 2 dimensions. So it's important to remember that, because otherwise we can start to make some, let's say, biological interpretations where it may not be appropriate to do so. The other thing to realize here, is that we can in this case see all these dots that are labeled by the cluster they belong to. The clustering is done by flow sum. That's a different algorithm. So the actual grouping of cells together is done using an algorithm that uses all of the dimensions. And then we simply plot that data in 2 dimensions using that. So they're 2 separate algorithms that we use to kind of work together in harmony, so to speak. That's basically what the fundamental stuff is. Then what we get to do is look at the cellular expression data and make some decisions about what cluster belongs to what cell type. So just as an example, one of the classics, Lysis G, which is always highly expressed on mouse neutrophils, is kind of on this little island at the top. So we would infer that cluster 8 is mouse neutrophils, and we can annotate it that way. And we can basically go through this entire process by annotating clusters based on what we know about their biology. The nice thing here is that every cell gets a label. So even if we come across a cell type where we're like, I have no idea what that is, or I don't know what to call that, we can still give it a label. We could call it unknown, or we could just leave it as the cluster number. It doesn't really matter. We get to call it something, so that way we can still quantify it, even if we don't have a great idea of exactly what it represents. Once we've annotated, this looks a little bit more interpretable. These are cell types and labels that we're familiar with. Once we've annotated, it's then reasonably trivial just to quantify that data. Once we've quantified it, as you'd imagine, we get plots that are actually pretty familiar. Let me just zoom out a tiny bit. So once we understand what the cell types are in our data set, we basically just ask the computer to calculate everything about each cell type in each sample. So how many cells are there, what the proportional differences are, if any of the cells are expressing a certain level of a chosen marker. All of these things can be calculated and then plotted automatically. And so we very quickly get from kind of our raw data, we take it through the clustering, through the annotation, and then through to some meaningful statistical analysis. This is by no means the end of the statistical analysis. You may go on to do kind of more complex things, but this is really just designed to give you a simple set of comparisons between your experimental groups so you get very quick answers out. And it may not be statistical tests that are like the most suitable for your assay. You may want to go and kind of do other things or do this in Prism if that's your preference. but it gives you at least a starting point where you get to look at where your kind of major differences are. So that's the overview of the workflow. Once you've kind of gotten to grips with like installing and loading packages and interacting with the R code, you'll find being able to work through a process like this is actually fairly straightforward. And it kind of becomes much easier once you're familiar with that process. If you're then going to go into something a little bit more complex or a bit more comprehensive, we have other options here. This next one here, discovery workflow with batch alignment, that's what we're going to tackle in the advanced workshop for anyone who's coming to that. Essentially what we're going to do here is use a tool called Cytonorm to adjust for batch technical batch effects that happen in two data sets. The idea here being that if you stain two sets of samples on 2 different days, you may have slight differences in staining levels. And so, how do you know what's a biological effect versus what's a technical effect? Using some of these tools, we can make that assessment and actually correct the data so that we remove technical batch effects, leaving only the biological differences, which is what that workflow will tackle specifically. We also have on here a range of tutorials, various aspects, either to do with like, let's say, technical aspects of like, how does the clustering work? How do you pick which markers you want to work? Various aspects like how you pick transformation values for arc-sine transformation. But then some of this is also a bit more immunological about sort of how we're making some interpretations about what's happening. So those are all really good to check out if you're kind of getting started and trying to work through some of these things, which is very helpful. So if we've still got time in the workshop. So if anyone is struggling in particular with like installing or running stuff, we would love to hear. Well, we wouldn't love to hear that, like that's a shame, but we would love to have the chance to help you out. If you are keen to try to move beyond just the script we provided for today's workshop, then by all means, absolutely give a try at some of those other scripts, those other workflows. We're here to help. We'd also love to hear about what you're attempting to do with these analysis approaches in your own work. We're very interested to hear what you're up to. So we'll stay on the line. Feel free to try out some more of these things. If you want to go to the website where we linked, then please feel free to do so. Give any of this a shot and let us know if we can help. Yeah, for your convenience, we've just dropped in links to both of those workflows that we mentioned. It's just a folder with the data and the script kind of pre-set up. So that's a little bit easier, just to go there directly and download it. They're also on the virtual computers for anyone using those. They're kind of pre-installed on the desktop on the virtual computer. So feel free to have a look at those if you're using one of those computers. If you're having some issues loading the data, I mean, you can share your screen if you like. If you feel like it, we can kind of look at what's going on specifically. Otherwise, if it's a problem where like it's physically not importing the data, like you're running the line that should, and it's not doing it, most of the time, something before that has gone wrong, and you can kind of just start again from the top. That's usually the most straightforward way, because I don't know, it's like somehow the directory has changed and like it happened without you noticing. That happens to me all the time. And if you go from the top, you can kind of just start again, and that should fix it. If that's not a solution, if that doesn't work, then like we can have a look and give you a hand sorting it out.
Yeah. Can I just share my screen just in case? Yeah, I'm doing everything right. Yeah.
Yeah, I'm not sure, but yes. Let's make sure the volume's high enough.
Yeah, I think I load the pack. I'm not sure. I run everything, but I downloaded the CSV files, but yeah, I don't actually know how to load the files.
Yeah. These read files, is that one of the Spectre functions?
Yeah,
so hang on. If you, what you should try running, is this on the VRD or is this your own computer?
This is your own computer.
Sorry, what?
Yeah. Sorry, actually.
Is this on the VRD or is this on your own computer?
On my own computer.
Yeah. So it looks like we want to double check that you've installed Spectre. So
yeah. So the package.
It can't find a function. Generally, it means that either the package that uses the function hasn't been installed, or it may not be open yet.
Okay. So should I do the library?
Yeah.
The library one.
And read file.
No. Spectre.
Yeah, so just if you type Spectre with a capital S there, it should see if it's already installed.
Yeah, there you go.
There we go.
Oh, missing a C.
Oh yeah, right. Oh yeah, if you try the line again, yeah, it's missing a C in
Oh,
Spectre.
sorry. Yeah.
Yeah. Number of times I went for the American spelling by accident. Just like, oh man. Brilliant. It's really unintuitive. When it works, it doesn't tell you anything.
Yep.
If it doesn't work, it tells you something. So if you try that line again, at this point, that should work.
I tried again, I think. Is it correct?
So when you're in console, you can click. Also, try running the read files.
Yeah, well, run it again. I think he meant run the read files again.
So,
Yeah, I think probably, I mean, if the library is now loaded, probably the easiest way is you can basically just take the script from the top again. Because I think, well, actually, there's two things you can do. Let me get my little annotate thing going here.
yeah,
So you could, if you want to, I mean, it just makes it a little bit easier, but you can hit that button first, the little like broom. Is it broom?
Yeah.
It's called a broom. And that'll just kind of delete everything that we've done. It won't delete the script. It'll just delete anything we've kind of run.
Delete all the variables.
Yeah.
okay.
And so that's often good just because
Okay. Yeah.
going from the start, so to speak.
Yeah, sure.
So if you then start running the code from the top, so select a section of code, hit Command Enter, or
Should I do the source or just select?
no, just do it little bit by little bit.
Highlight line by line and then do run selected line or alternatively.
From here or?
Yeah. So if you click on the line, and then you can either highlight it all, or you can just put just click on the line somewhere and then click Run, and it will run that line.
Okay,
I
sorry.
think
I think I did the wrong. Yeah.
If you hit Enter, it'll just like remove the line because it's treated.
Yes. Sorry, I did the wrong thing here. Yeah. I'll try to figure out myself first. Yeah, but.
my scribble. Nice. Looks good.
Yeah.
Yeah, and pretty much
Yeah, I just run through
that's to
everything.
your heart's content.
Yeah, there's one trick. If you want to run one whole line, you don't need to highlight the whole line. You can just put a cursor on that line and then push run. You can do that as well.
Okay, like that.
So for instance, on line 39, just yeah, you can just do that as well.
Okay, yeah, right.
Oh yeah, that update thing.
Yeah, that happened again. So just put your cursor on the console and just press enter.
Yeah, if you click down here and then backspace over library, you'll need to just type in A and hit enter. Yeah. Or oh, probably should have gone for A. My mistake.
Don't worry.
It's fine. It'll update a couple of
Yeah,
packages. Yeah. I never got to figure out why it's missing. So when we install it and Fitsny doesn't run, there's a particular like Windows config thing that like for some reason doesn't come up on the virtual system, but it does on the VRD. And I never got to I Googled it a bit, but no. Yeah, it was really
They
weird.
did have some instruction on the GitHub on how to install it.
Like it's
We have
It's not the same way.
Yeah.
It's always installing packages that seems to take the longest.
Yeah, if you want to cancel it, there is a stop button.
Nah, it's
Okay.
You can do that, but
well.
just leave it.
Okay, I leave it. Yeah.
It's too risky. It's like, you know what, you don't want to anger the Argos. Kind of
Yeah,
thing. I mean, the nice thing though, of course, is even if R quit or it crashed or something, the nice thing with the code structure is because it's by definition reproducible, and so even if your computer crashed right now and you get back to it, you could just run the same code and you would get the same result.
Okay, yeah, good to know that. Thanks.
Okay, there we go.
Yep. Okay. So, all right. Yeah. So it should be okay now, right? If it's,
Yeah.
Okay. I'll just try again. Just run through everything on my own. Yeah. But thanks for that. Yeah.
Our pleasure. If you have any other issues, feel free to share the screen again. We'll give you a hand tackling it.
Yeah. Thank you so much.
Ian, that's an interesting problem. I don't know why I would be doing that, but I'm glad to hear that worked. It's always something. It's always something with Windows, isn't it? Cool. And yeah, so I mean, if I don't know if you come across anything which doesn't work properly or anything that you have any questions about, please do feel free to reach out. Or any objections. That's the other thing. like feedback, which is like, I don't like how this is run, is just as valuable sometimes because we can kind of figure out what things don't make sense. So even if it's like, I didn't understand this bit of code, we would actually genuinely love to hear that feedback. It helps us to improve things as well. But otherwise, I hope it goes well. So we're at 6 o'clock Sydney time, at least at the moment. So if I will stick around for a little bit in case anyone wants to ask any questions or sort out any problems. So we'll stick around for a little bit. Otherwise, I guess that's the kind of formal end to the workshop. So please do feel free to keep on with any of those resources. All of that stuff will remain online and you'll get access to the recordings after the fact as well. So please feel free to keep on with it. We'll stick around for 5 or 10 or so before we close out. So thank you for coming and we hope that it's been helpful. Feel free to reach out with any questions or queries about anything.
Hey Thomas, Osvaldo here. Can I ask something?
Absolutely.
I'm not sure if I'm up tomorrow for the advanced course. And I think that I did not apply for it because I thought, well, maybe it's way too advanced. So, but I think that it might be suitable for it. So is there any chance that I could assist or I could be included on it, or is this not possible?
I can get you in. I know a guy who knows a guy, so that should be fine.
Fantastic. Thank you for that. Appreciate that.
Absolutely. I mean, it's certainly also good to have a bit more kind of, I suppose, inbuilt interaction too. So that can be very helpful.
Perfect. Thank you, guys.
Oh, thank you very much. We really appreciate it.
Yeah, thanks, Giovanna, and thanks, Thomas. Very, very useful. Everything running smoothly now, so I just need to get my head around how it all works and play around with it a bit, but lots of fun to be had. So thank you very much, and catch up with you guys tomorrow.
Yeah, definitely. Thank you. Thank you so much for coming. I hope it's been helpful. So we'll probably end the Zoom session there. Please do feel free to get in touch if you're struggling to get anything working, and otherwise all of that content will be online as well. Thank you everyone for joining us.
See ya.